Welcome to our second video on hormones. In this video, we'll discuss the primary anabolic hormones and how we can program resistance training to take advantage of it. This information comes from Chapter 4 of Essentials of Strength Training and Conditioning and is the second half of the chapter. In the previous video, I explained what hormones are and how they interact with their target tissues. We discussed second messenger systems, their prevalence in the blood, and the importance of receptors for hormone target tissue interaction. Briefly, here are some examples of adaptations in your endocrine system. 1. Altering the synthesis and storage of hormones. 2. Changing the time needed for clearance of hormones through the liver and other tissues. 3. Modifying the receptor count and receptor type in the target tissue. 4. Changing the contents of secretion from various glands. 5. Altering the degree of interaction with cell nucleus. The hormonal response to exercise is highly adaptable. To help explain this with each of the primary hormones, we are going to use resistance training as the exercise example. This is despite training studies on the hormonal effects on aerobic and anaerobic training. The first anabolic hormone that we're going to discuss, and the one that probably comes to mind when you hear anabolic, is testosterone. According to the literature, the following exercise variables can increase serum testosterone concentrations. Exercises that focus on large muscle groups, such as deadlifts, squats, bench press, and chin-ups. These multi-joint movements create high degrees of mechanical tension, stimulating the musculature through its full range of motion. Activating a large amount of muscle mass increases serum testosterone content. Smaller exercises like bicep curls or single leg knee extensions may increase serum testosterone content slightly, but large muscle mass movements have the most significant impact. Another one is utilizing heavy resistance, in particular around 85 to 95% of 1RM. And this has been shown to be the best loadings for increasing acute endogenous, that is natural, testosterone concentrations. However, it's important to acknowledge that at certain training phases, we can't train at these high intensities of RM. A potential example could be when we are training for hypertrophy, where we are using high repetitions and a subsequent lower percentage of RM loadings. Another one is implementing a moderate to high volume of exercise and varying the types of exercises. Different exercises activate different motor units and stress muscle fibers in unique ways. Another way is using short rest intervals, which can increase testosterone concentrations. But be aware that short rest periods may also reduce your ability to train with heavy loads. Another one is having at least 2 years of resistance training experience, which can lead to a greater anabolic effect after training. It's worth noting that heavy resistance training with low volume, that's around that 1 or 2 repetition range, may not change testosterone concentrations in your blood, but it could potentially increase the number of receptors in the muscle tissue, making more binding sites available for testosterone. In summary, large muscle mass exercises with adequate total work volume result in greater acute increases in total testosterone concentrations. Most of the research has been conducted on men, and males typically experience a larger increase in testosterone following training than females. However, testosterone is still essential for female athletes' muscularity and strength. Changes in muscle size and strength in females may be more related to estrogen and potentially human growth hormone than it is in males. Testosterone exists in 2 forms, free testosterone and testosterone bound to sex hormone binding globulin, otherwise known by the acronym as SHBG. Free testosterone accounts for 0.5 to 2% of total testosterone. To illustrate this, a study that looked at the response of heavy resistance exercise, where the participants performed 6 sets of 10 repetitions at 80% of their 1RM, acutely increased their free testosterone in both men and women participants, despite a smaller increase in the female participants. A note here is that being able to perform 10 reps at 80% of your 1RM 6 sets is quite tough. However, as this study showed, this type of intensity was able to elicit this acute positive increase in testosterone concentrations. Women have about a 15 to 20-fold lower concentration of circulating testosterone than men, but it does vary substantially between women and subgroups of female athletes. The difference between men and women can be highlighted by this figure, which is the result of the squat study. This graph displays the total testosterone response in men and women, with the men's data at the top and the women's at the bottom. Notice the dramatic difference. We have pre-training, mid-training, immediately post, and also data points at 5, 15, 30, and 70 minutes post-exercise. Now, this is after 6 sets of squats with 80% of 1RM, with 2 minutes of rest in between. One thing to point out is how long testosterone is elevated for. The second graph shows the free testosterone response to this same workout. Notice here that the free testosterone response is much greater in magnitude over the resting values in men. For the females, there is just the slightest increase. So what this is showing us is that after exercise, testosterone increases in response to the demands of the exercise protocol. And then the receptors will either increase their binding availability or they'll do nothing because perhaps there's a lack of a signal to increase the muscle-related metabolism. Remember, it's both the free testosterone circulating in the blood and the receptor availability on the muscle that will control the hormone to target tissue interaction. But there are also other factors at play, not just testosterone. There are also nervous system regulators, satellite cells at play, and these may be more affected at certain time points in training depending on the window of adaptation available in the target tissues. So it's just not testosterone that is driving the anabolic response. There are other physiological factors as well at play. The next primary anabolic hormone to consider is growth hormone, which is particularly important for the normal development of a child. Growth hormone is secreted by the pituitary gland and has effects all throughout the body. Multiple factors can affect growth hormone secretion, such as stress, fitness, diet, age, gender, body fat, exercise, and also injury status. Growth hormone plays a vital role in adapting to the stress of resistance training. There are a few primary physiological roles of growth hormone and its related compounds. In particular, increases in growth hormone concentrations has been shown to decrease glucose utilization, increase protein synthesis, collagen synthesis, stimulate cartilage growth, enhance immune cell function, and increase fat breakdown. Briefly discussing each point. Firstly, growth hormone decreases glucose utilization by the body, which can lead to increased blood glucose levels. Growth hormone secretion is pulsatile, while with surges in secretion occurring after the onset of deep sleep that are especially prominent at the time of puberty. In normal subjects, growth hormone secretion increases in response to decreased food intake and to physiological stresses, and decreases in response to food ingestion. In the state of feast and sufficient nutrient intake, where insulin is increased in the liver and insulin growth factor 1 production is stimulated, growth hormone promotes protein anabolism. Whereas in a state with decreased nutrient intake and during sleep and exercise, the direct effects of growth hormone are more predominant. And this is mainly characterized by stimulation of lipolysis. Growth hormone increases protein synthesis, which is important for building and repairing muscle tissue. Growth hormone stimulates its effects directly on target organs and cells interacting with growth hormone receptors and through stimulation of insulin-like growth factor 1 production. Growth hormone plays important roles in regulating somatic growth and the metabolism of carbohydrates, lipids, and protein. Growth hormone secretion is stimulated by growth hormone releasing hormone, GHRH, and is inhibited by a regulatory hormone called somatostatin. Growth hormone stimulates cartilage growth, which is important for bone growth and development. It stimulates the proliferation of chondrocytes, which are the cells responsible for cartilage growth. Growth hormone also increases the production of extracellular matrix components, which are important for cartilage growth and development. It enhances immune cell function, which helps the body fight off infections and diseases. It also increases the production of immune cells such as T cells and B cells, and enhances the activity of immune cells by increasing their proliferation and function. Growth hormone secretion is also affected by the type of resistance training protocol, particularly the rest period. Short rest period workouts have been shown to result in greater serum concentrations compared to longer rest protocols. However, it's important to note that the increase in growth hormone doesn't necessarily mean a direct correlation to muscle growth. There's still much to learn about the specific relationship between growth hormone and muscle hypertrophy, but it is clear that it plays a role in the overall adaptation to resistance training. There are different forms of growth hormones in our body. One of these forms is called the bioactive growth hormone, and the other is a smaller version called the 22-kDa monomer growth hormone. When we look at the levels of the bioactive growth hormone, when we're resting, not exercising, we notice that these levels can change. Specifically, there are some versions of this hormone that are larger. In other words, they have a higher molecular weight than that 22-kDa. That might show these changes. However, even with these observations, scientists are still trying to understand how doing resistance training over longer time periods can affect the levels of growth hormones in our body. With respect to the growth hormone responses in women, it has been shown that they have higher blood levels of 22-kDa growth hormone throughout the menstrual cycle than men do due to the greater frequency and amplitude of secretion. 22-kDa is an isoform of human growth hormone and is secreted from the pituitary gland under hypothalamic control. The secretion of 22-kDa growth hormone is classically stimulated by growth hormone releasing hormone and is inhibited by somatostatin. Despite the fact that the mechanisms underlying this variation are not fully understood, hormone concentrations and hormone responses to exercises vary within the menstrual cycle. Research has shown that women had 22-kDa growth hormone concentrations at rest that were significantly higher than those of men during the early follicular phase of the menstrual cycle. Furthermore, 22-kDa growth hormone concentrations did not rise above resting levels when a heavy resistance exercise protocol categorized by long rest intervals, that's 3 minutes, and heavy loads of 5 RM was used. The levels of serum 22-kDa growth hormone, however, significantly increased when a shorter rest period, that's 1 minute, and a more moderate resistance exercise protocol was used, and that was changing it to a 10 RM. Due to the changes in the resting levels, hormonal response patterns to various resistance exercise routines may change over the course of the menstrual cycle. In addition, hormonal contraception, such as estrogen-containing birth control pills, appears to increase the 22-kDa growth hormone response to resistance exercise. During resistance training workouts where they used short rest periods, resulted in a higher 22-kDa growth hormone increase compared to longer rest periods. However, the response patterns of men and women to the same resistance exercise protocol are comparable. It is interesting to note that resistance training has been shown to change bioactive growth hormone in females. While the 22-kDa form showed little change, it has been reported that the resting concentrations of growth hormone can increase. Additionally, older women have lower bioactive growth hormone concentrations than younger women, and resistance exercise stimulates bioactive growth hormone more effectively than endurance exercise. Additionally, it has been found that stronger women have higher levels of bioactive growth hormone, supporting the significance of the higher aggregated forms of growth hormone. These recent discoveries regarding growth hormone serves as another example of the endocrine system's potential complexity and serves as a reminder that there is still much to learn about these important signals to our tissues. To determine whether changes occur within resistance training, growth hormone concentrations need to be measured over longer time periods post-exercise session, up to 2 to 24 hours. The area under the time curve, which includes an array of pulsatile effects, tells whether changes in the release have occurred. Resistance training's effects on growth hormone concentrations hasn't been extensively studied, but observations of elite lifters' normal single measurements of their resting 22 kDa concentrations suggest little change. Resistance training-induced growth hormone adaptations are probably mediated by various feedback mechanisms: alterations in receptor sensitivity, insulin growth factor potentiation, diurnal variations, and maximal exercise concentrations. The typical trends for training-related changes in growth hormone appear to be a reduction in 22 kDa growth hormone response to an absolute exercise stress, and alterations in 22 kDa growth hormone pulsatility characteristics. The reduction in the 22 kDa growth hormone exercise-induced responses with training may be an indication of possible interactions with other molecular weight forms. For example, in a group of elite weightlifters over a 9-month period, individual responses were extremely variable, with no discernible changes in the group over time. This likely means that higher amounts of aggregate growth hormone were being produced. Currently, it seems that training primarily affects the 22 kDa growth hormone's exercise-induced responses, with only minor menstrual phase increases and decreases affecting resting concentrations. Growth hormones and their effects are conducted primarily through small polypeptides termed as insulin-like growth factors, or alternatively, somatometers. It's a widely accepted hypothesis that an insulin-like growth factor superfamily might serve a significant role as a biomarker for health and performance. Examining these growth factors more closely, we find that insulin-like growth factor 1 constitutes a 70 amino acid polypeptide, and IGF-2 consists of a 67 amino acid polypeptide. The latter's function is not as well understood. Accompanying these are a superfamily of peptides and their binding proteins. The process of insulin-like growth factors secretion is initiated in the liver, stimulated by the 22 kDa growth hormone, which triggers liver cells to synthesize IGFs. The regulation of insulin-like growth factors is not solely dependent on growth hormone. Other factors such as thyroid hormone and testosterone are also involved in this complex regulation. Both growth factors, similar to numerous polypeptide hormones, are initially synthesized as larger precursor molecules. These precursors then undergo processing to form the active hormone variants. Insulin-like growth factors, post-synthesis, travel in the blood in a bound state to binding proteins. Upon reaching their target tissue, insulin-like growth factors dissociate from the binding protein to interact with the receptors. Blood levels of insulin-like growth factors are typically measured either as total levels, inclusive of bound and free, or as free insulin-like growth factor concentrations. Regulating the amount of insulin-like growth factors available for receptor interaction is an important role played by at least 6 different circulating binding proteins, referred to as IGF binding proteins 1 to 6. The response of binding protein 1 and binding protein 3 to exercise has been studied extensively. Each binding protein responds to exercise-induced stress independently, each exerting its unique biological interactions. The role of binding proteins extends to the transport and physiological mechanisms of insulin-like growth factor. In a feedback loop, insulin-like growth factors have been demonstrated to stimulate the secretion of its own binding proteins from within the muscle cell, thus modulating the cell's responsiveness to insulin-like growth factors. The circulating insulin-like growth factor binding proteins restrict access of the IGF peptides to receptors, and their levels are influenced by 22 kDa growth hormone concentrations. Other influential factors include nutritional status and insulin levels, which have been proven to be important signaling mechanisms for insulin-like growth factor release. The nutritional status plays a substantial role in insulin-like growth factor transport, production, and regulatory control, thereby affecting its cellular interactions. Variations in nitrogen balance, protein intake, and overall nutritional status can impact numerous mechanisms. Binding proteins seemingly act as a reservoir for insulin-like growth factor, and their release is signaled by the presence of a receptor on the cell. This mechanism extends the viability of insulin growth factor and could theoretically curtail the degradation of insulin-like growth factor. In the context of strength training, these mechanisms are influenced by exercise-induced stress, acute hormonal responses, and the need for remodeling of muscle, nerve, and bone tissue at the cellular level. The interactions between multiple hormones and the receptors serve as powerful adaptive mechanisms in response to resistance training, leading to changes in muscular strength and size. The primary insulin-like growth factor studied in the context of exercise is IGF-1, due to its significant role in protein anabolism. That is, the process of building up proteins in the body. IGF-1, like IGF-2, belongs to a superfamily of polypeptides and binding proteins. Polypeptides are chains of amino acids, and binding proteins are proteins that act as carriers. Both these entities play vital roles in health and performance. The reason for acute or sudden increases in blood levels of IGF-1 are not fully understood. However, they're probably related to the disruption of various cells, including fat and muscle cells, because these cells manufacture and store insulin-like growth factor. IGF-1 exists across different biological compartments such as blood, interstitial fluid, that is the fluid that fills the spaces between most of the cells in the body, and also muscle. There have been studies to understood whether the circulating IGF-1 responses to exercise accurately mirrors what's happening in local fluids surrounding the muscle. One study involving both men and women found that the increase in circulating total and free IGF-1 was not correlated with increases in interstitial fluid IGF-1 concentrations or muscle IGF-1 protein content. This suggests that exercise-induced increases in circulating IGF-1 might not accurately reflect the local IGF-1 signaling taking place, emphasizing that circulating concentrations are only one aspect of the endocrine response patterns. It generally takes between 8 to 24 hours for insulin growth factor to be produced and released from the liver after being stimulated by growth hormone. This suggests that IGF might be released from storage sources other than the liver. The release might be due to the disruption of cells already containing IGF, or the growth hormone mediated release of IGF might have a different time course in response to certain types of exercise compared to injection response studies. Changes in the circulatory concentrations of IGF in response to different types of exercise protocols appear to be closely linked to the regulatory factors of IGF release and transport. To assess the specific effects and relationships to growth hormone in serum, it's necessary to evaluate serum changes over longer periods. In both women and men, IGF-1 levels have remained stable despite improvements in strength and power over a 2-month training program. Exercise-related increases in IGF-1 seem more likely if resting concentrations are of a lower value, such as 10 to 20 nanomole per liter. Thus, the stability of resting concentrations might be influenced by the absolute amounts of IGF-1 in circulation. With regular training, higher resting concentrations such as 35 to 45 nanomole per liter could make acute exercise-induced increases less likely. Importantly, free forms of IGF-1 that aren't bound to any binding protein might be the key elements influencing target tissues, particularly skeletal muscle. IGF-1's influence on muscle may primarily be due to its autocrine and possibly paracrine release mechanisms. Autocrine refers to a mode of hormone action where a hormone affects the cells that produces it, while paracrine refers to the actions where the hormone affects nearby cells. At rest, fat cells contain relatively high concentrations of IGF, while skeletal muscle contains very little. However, mechanical stimulation, overload, and stretching of muscle cells during resistance exercise causes them to significantly increase their production of IGF-1. This IGF-1, often referred to as mechanogrowth factors, exerts autocrine functions. Scientists believe that different versions of IGF-1, called IGF-1 splice variants, might play a role in how muscle cells grow and become specialized. This growth might be influenced by the mature form of IGF-1 and not by something called E-peptides. E-peptides are sequences of amino acids that are derived from the processing of the insulin-like growth factor 1 precursor molecule. When IGF-1 is produced, it's initially part of a larger precursor molecule. As this molecule is processed, the mature IGF-1 is released, and the remaining parts, which are the E-peptides, are cleaved off. There are different E-peptides depending on the specific splice variant of the IGF-1 precursor. These E-peptides have been the subject of research interest because they may have biological activities of their own, separate from the actions of mature IGF-1. Some studies also suggest that E-peptides might play roles in cell growth, differentiation, and other cellular processes. This topic is still being studied and is of great interest. While IGF-1 has been shown to be responsive to exercise in some studies, this doesn't always follow a classic endocrine response. If we look at the training adaptations of insulin-like growth factors, the responses of IGF-1 to heavy resistance training can be variable, but studies have shown that the changes are based on the starting concentrations prior to training. To put it simply, if basal concentrations are low, IGF-1 increases. If they're high, there's either no change or it decreases. It's also worth noting that the intake of food or the level of caloric restriction, or even both, influence the resting and exercise-induced concentrations in the blood. Research has shown that in women that resistance training can increase total IGF-1 and decrease IGF binding protein 1 concentrations during acute resistance exercise. These findings suggest that exercise mode-specific adaptations in the circulating IGF-1 system can indeed occur. However, the effects of training on the IGF superfamily response patterns are still a work in progress, encompassing many different aspects. Similar to growth hormone, training-induced adaptations in IGF-1 are likely reflected in a variety of mechanisms related to the type of IGFs, their release, transport, and receptor interaction. Moreover, the interaction with other anabolic hormones cannot be overlooked, as these often target the same outcome, such as protein synthesis.